All right. So with DNA, okay, they're going to ask you things. They're going to focus on the structure. How does DNA work? Why does DNA have a certain 3' to 5' orientation? How does that relate to organizing the molecule? What makes it stable? So I'm going to hit a couple of these right now with you guys. And the next period, we'll get into RNA and the differences between DNA and RNA. We'll talk about why prokaryotes and eukaryotes are different and all that. But as far as like where DNA was first discovered, 1944, this group, Avery, McCarthy, and McLeod, they're the ones who determined that DNA was actually the genetic material. They looked at this experiment that this guy Frederick Griffith did with mice, right? And basically what he did with this is said, I had mice and he injected them with virulent bacteria and they died. And then he injected other mice with non-virulent and they lived. Then when he mixed the virulent and non-virulent, right? And what he did is he killed the virulent bacteria that were deadly. When he mixed them with the non-virulent, all the bacteria killed the mice. And so he came up with this idea that there was some kind of transforming principle. There was something that was being transferred from the dead bacteria to the live bacteria. But he had no idea what it was, right? But he knew it couldn't be protein because heat didn't destroy it, right? So these guys in 1944, they did this experiment where they isolated all these different components from the cell. And they just systematically, they did a process of elimination. They said, let's get rid of the protein. Oh, they still transform. Let's get rid of the carbohydrates. Oh, it's still transformed. Let's get rid of the RNA. Oh, it's still transformed. Okay, let's get rid of the DNA. And then nothing happened. And they were able to conclude from that you needed DNA to transform the bacteria, right? So in 1944 is when we actually figured out that DNA was the genetic material, right? And then in 52, Hershey and Chase did the blender experiment with viruses to confirm that whatever viruses were injecting into cells was DNA, and that was changing the genetics of the cells. And they determined in 1952 that DNA was what was being transferred from the virus into the bacteria, right? And that was what was causing changes in the bacteria, right? And then our good friends Watson and Crick, they had the whole structure of DNA. They built the model based upon Roswell and Franklin, right? Franklin did the whole X-ray crystallography stuff, and these guys figured out the structure, right? So Watson and Crick, 1953, they published the structure, right? And we know the structure, it's a double helix, right? So you got this double helix, right? It's 2 monomers of nucleotides, okay? Make sure you guys know your nucleotides, what they look like, right? You have your phosphate, the sugar, and the base, right? The strands are anti-parallel. I know I talked about that last week, right? One side runs 3 to 5, the other runs 5 to 3, right? They're opposite each other, right? And the whole purpose of that is so that you can get the bases to pair up with each other, right? Remember, the bases form like the rungs of a ladder, right? And the phosphates and sugars are the backbone. And it's this sugar phosphate backbone that makes it really stable, right? And then the bases pair up, okay, with hydrogen bonds, right? Hydrogen bonds are weak, okay, but these are how the bases pair, AT, CG, okay? And the bond that's in the backbone, that's really stable. Do you remember what that's called? It's this bond. It's between the phosphate and, yeah, the phosphodiester. Yeah, exactly. Yep. Right? That's very stable, okay, a phosphodiester bond. Okay? So that's what gives DNA its stability, right? And then the hydrogen bonds in between give it stability too, right? The AT double bonds and the CG triple bonds that are hydrogen bonds, this also stabilizes the molecules. But at the same time, they're hydrogen bonds, so what does that mean about them? Is that a strong or a weak bond? Individually, weak. Collectively, very strong, which means it's easy to break those bonds, okay? And so even though this stabilizes the DNA, it's still easy to break for replication, right? And that's one of the things that Watson and Crick figured out, was that those hydrogen bonds are weak and it's easy enough to get in there and separate those, right? And so we know that that's how the DNA gets separated, okay? But again, the basic things, your phosphate sugar base, bases pair up with each other. On the inside, they're anti-parallel. There's directionality to the DNA, which is important when we get to replication, okay? And that's something to focus on. And you got that double bond, triple bond situation here with the hydrogen bonds, which gives you more stability. Eukaryotic DNA, we have multiple linear chromosomes, right? What is the DNA surrounded by? It's in the nucleus, but what does the DNA attach to and coil around in eukaryotes? And it gets very clumpy. So what causes our DNA to get super coiled and clumpy and it's bound to these things? It's actually up there. It's DNA with proteins, right? Our DNA is associated with these proteins, right? Those proteins are called histones, right? Those histone proteins regulate DNA. They regulate the genes, right? The DNA plus protein is given a name. It's this collective grouping of the DNA and the protein, and we only find it in eukaryotes called, starts with a C, chromatin. Do you remember that? Right? Chromatin. This is only in eukaryotes, okay? Only eukaryotic cells have DNA supercoiled and wound up around proteins, okay?
So wait, chromatin is the one that attaches to on the kind of the X basis, or is it histone?
Histone. So histones are the proteins that bind to the DNA, and then when that DNA and protein is together, that association is known as chromatin, right? So the coiled up DNA wrapped around the proteins, that's chromatin, right? And so those histones, the individual histones, they're going to bind to the DNA at gene sequences, and they control gene expression, right? Remember when I showed you guys the lick your rats thing, right? And when the mom licked the rat pups, the DNA uncoiled, right? What the DNA was uncoiling from was the histones. It was releasing it from the proteins so that it was opened up a bit, okay? So only eukaryotes have these histone proteins that the DNA coils around, right? Prokaryotes, the DNA is just singular. It's a single chromosome. It's still a double helix, right? But it just floats in the cytoplasm, okay? They don't have any protein associated with their DNA, okay? So unlike our cells, eukaryotes, plants, animals, protists, fungi, right? Where we have the DNA supercoiled around all this chromatin, prokaryotes, they just have that one single round chromosome, okay? With no protein. Why do we need to supercoil and compact our DNA down? It's a practical matter. Yeah. It actually decreases the surface area, right? By compacting it down, it gets supercoiled and condensed so it can fit, right? Each cell in our body, remember, has 6 feet of DNA. So to get 6 feet of DNA to fit in the nucleus, it has to be supercoiled. So it can be condensed down and it can fit in the nucleus. That's really the purpose for that, right? So by supercoiling our DNA, it can fit, and it's wrapped around the proteins which help to condense it down, right? So we have chromatin, prokaryotes don't, okay? And the other thing that prokaryotes have is plasmids, right? That extra piece of DNA, right? The extra chromosome of DNA, right? And that helps them because that gives them extra genes, right? Gives them a little bit more variety so that they can swap alleles with each other, okay? Let me just show you a quick picture. Let me just pull this up. And then we'll take a break. Let's see. Again, these are all my little review videos that I made from a while back. Yeah, here we go. Okay, right here. So these are the histones, okay? This is right out of our notes. And here's the DNA. And the DNA coils around that, and it supercoils, and that's what forms the chromosome, right? And that association of DNA coiled around the DNA, that's chromatin, okay? Only in eukaryotes do we have chromatin. Okay, prokaryotes don't. And then these histones are important in gene expression, which we'll talk about next week, where certain chemicals can cause these histones to dissociate from the DNA, and it opens it up, and that's how we can access genes. Okay, and so we can use things like methylation and acetylation, which can control the coiling of the chromosomes, and it allows us to have access to the genes or not. Okay, and then the other thing on this, since I have the picture up, and I know we looked at this a little while back. Yeah, here we go. Right, the 3' 5' orientation of the DNA. One side of the helix points up, the other side points down, and then there are your hydrogen bonds. Right, your CGs have 3 bonds, your ATs have 2. So if DNA has a lot of CG interactions, what does that do in terms of its stability?
More stable.
Yeah, more stable, right? It's more heat resistant. So a lot of bacteria that live in hot springs or hot environments, they notice that they have a lot more CG bond interactions, which makes their DNA a little bit more stable, a little bit more heat resistant, right? When you heat DNA up, all it does is it separates. And then when you cool it, it comes right back together again. Good.
Wouldn't that make it kind of like a coded different bacteria, because it would have to kind of transcribe a bunch of stuff that would change from like A and T to G and C?
Yeah. So what actually happened is, like, over time, like, they noticed that these bacteria, they have different sets of genes and different sequences that code. Some of the stuff codes for the same stuff, some of it doesn't. But they basically are different from other bacteria. Like, you could take like they look like 2 similar species, but genetically they're very different because the heat resistant ones are going to have different sequences. So I guess it's like, okay, you're in a hot environment. If you have more CGs, you're more likely to survive, right? And so over time, that's what got passed on. And so you see that, as a more predominant one. And yes, some of those bacteria have really different proteins, right? We actually use one of the proteins for PCR from some Thermus aquaticus because it's got a much more resistant, heat-resistant DNA polymerase. Okay. All right, let's take a break. When you come back, we'll talk about replication. We'll get into RNA. I'm going to stop this. Yep.
I'll see you next week.
Oh, yeah, that's right. You got to run. Yep, go ahead. Yep, no problem.